Imagine taking a 1,000-page book, running it through like a heavy-duty paper shredder, mixing those pieces with a dozen other entirely different shredded books, and then...
And then trying to rewrite the original text perfectly from the scraps. Yeah,
exactly. It's just an impossible task. But that is exactly the scale of the problem scientists face when they attempt to sequence a tumor.
It really is. I mean, it's an incredibly chaotic environment. You aren't just looking at one pristine genome, right? You are looking at a collision of multiple dynamically evolving genomes all matched together.
And untangling that collision is our mission for you today. Welcome to the deep dive. We are looking at this landmark Nature Biotechnology paper today, which details this massive 7-year community effort known as the SMC Het Dream Challenge.
7 years. It's a massive undertaking.
It really is. And our goal for you today is to completely demystify this concept of single-sample tumor subclonal reconstruction. By the time we finish, you'll understand exactly how algorithms map out a tumor's evolutionary history from just one bulk DNA sample.
And we'll get into why this math breaks down in specific biological traps and what actually dictates the success of these models.
Okay, let's unpack this. Because setting the stakes here is important. Why does mapping this out even matter for a patient?
Well, it's vital because understanding a tumor's history is essentially mapping out a patient's clinical outcome. Tumors are not uniform masses, they are ecosystems.
Right, they evolve.
Exactly. High genetic heterogeneity, which just means a wide diversity of different subclonal cell populations within that single tumor, directly correlates to therapy resistance.
So if you hit a highly heterogeneous tumor with a targeted drug, you might, I don't know, wipe out the dominant clone.
Right, you wipe out the main one, but you leave behind a resistant subclone. And that subclone eventually drives a relapse. So we really have to know the evolutionary timing of these mutations to treat it effectively.
Okay, so since we are mapping this evolutionary family tree, we're relying on the usual suspects in the genomic toolkit, right? Like SNVs and large-scale copy number aberrations or CNAs.
Yeah, the single-letter typos and the big structural changes.
Right. So I assume the basic logic here is that because cells pass their mutated genome down to their daughter cells, algorithms can just track the variant allele frequencies, the VAFs of those SNVs, to trace the lineages.
Precisely. The VAF is key. If 2 different mutations consistently show up in, say, exactly 30% of the sequenced reads, the algorithm infers that they likely belong to the exact same subclone.
Because they're traveling together.
Exactly. They share a lineage. It uses statistical frequency clustering to group these mutations and build an ancestral family tree, showing which clone gave rise to which subclone.
But here's where I struggle with the physical reality of this. If we take a bulk biopsy like just a single physical sample, we are essentially listening to an orchestra of 100 instruments playing at once through a single microphone.
That's a great way to put it.
And we're trying to isolate the exact sheet music of, like, the third violinist. We've lost all the physical cellular boundaries when we extracted the bulk DNA. So how do the algorithms know they aren't just grouping random noise?
Well, that is the core vulnerability of the entire field. Because we sequence bulk DNA, we rely entirely on overlapping statistical distributions to reverse engineer the physical cells.
We're just guessing based on the math.
Yeah, basically. And because we don't have a time machine to actually watch the tumor grow in the patient, we have no absolute way to know if the family tree and algorithm outputs is actually correct.
Right, you can't exactly open the patient back up and count the subclones to check the algorithm's math.
No, you definitely can't do that.
Okay, let's do a quick recap here before we move on. First, tumors evolve into distinct subclones, like branches on a family tree. Second, we track them using those single-letter typos, the SNVs, and larger structural changes. And third, algorithms try to reverse engineer this history from a single bulk DNA sample using statistical frequencies.
That's the perfect summary.
So to everyone listening, think about this. If someone only saw the final result of a complex project you were working on, how accurately could they guess every single step you took to get there? That's what these algorithms are facing.
It's a monumental challenge, which is why, to truly validate these algorithms, the researchers had to build an arena where they completely controlled the truth.
Right, the SMC Het Dream Challenge. And this wasn't just like a quick software benchmark over a weekend.
No, not at all. This was a 7-year computational gauntlet. It involved over 12,000 algorithmic runs on massive cloud infrastructure.
12,000? That's wild. So how did they build this arena?
They had to create a ground truth. So they used a simulation tool called BAM Surgeon.
BAM Surgeon. Catchy.
Yeah, it's a great name. They started with BAM files, which are basically the standard files for normal, healthy DNA sequencing. Then, they mathematically injected simulated SNVs, insertions, deletions, and structural variants to create 51 simulated tumors.
So they basically played God with the DNA, but they didn't just make up random easy tumors, right?
No, they modeled them on real, highly complex genomic profiles from the PC-Topogy dataset. That's the pan-cancer analysis of whole genomes.
Ah, okay. So because they built these tumors digitally, line by line, they knew the absolute truth. They knew exactly which simulated mutation belonged to which simulated subclone.
Exactly. They created an unbreakable answer key. Then they invited teams from all over the world to submit their algorithms. 31 different containerized algorithms entered this arena.
Totally blinded to the answer key, I assume?
Completely blinded. And they were put through 7 distinct sub-challenges.
Yeah, and looking at these sub-challenges, it's really clear they weren't just grading the final output, they were grading the entire sequential pipeline.
Right, step by step.
Like sub-challenge 1A tested purity. What percentage of the bulk sample is actually tumor versus normal healthy tissue? Then 1B asks for the total number of subclones.
And 1C asks for the cellular prevalence, or CP, of the individual SNVs.
Right, and then it scales up, right. Sub-challenges 2A and 2B focused on clustering, basically grouping those mutations together. And finally, 3A and 3B ask the ultimate question.
Can you draw the correct ancestral family tree connecting all these subclones together?
The actual phylogeny.
Yes. And what's fascinating here is that testing the pipeline aspect is crucial. Subclonal reconstruction is basically a cascade of dependent probabilities.
Meaning if you mess up early, you're doomed later.
Exactly. If your algorithm miscalculates the tumor purity in step 1, it throws off the expected allele frequencies for every subsequent calculation. A 10% error in purity might completely erase a minor subclone by the time you try to draw the family tree in step 7.
Wow. Okay, time for another quick recap for you guys listening. The researchers built 51 simulated tumors with known true evolutionary trees. They tested 31 algorithms across 7 distinct tasks, ranging from estimating basic purity all the way to drawing the complex family tree.
It's the ultimate stress test.
So here's a question for you to ponder. Which of these tasks, estimating the basic purity of the sample or drawing the full family tree, do you think the algorithm struggled with the most?
The answer might actually surprise you.
Well, pulling this together, we have this perfect arena, all these blinded algorithms trying to reverse engineer evolution. Going into a massive study like this, you have to wonder where the actual bottleneck is.
Right, is it the biology or the math?
Exactly. Is it that tumors are just biologically too complex to map? Or is it that our math just isn't good enough yet?
That was one of the most revealing findings of the entire paper. They analyzed what actually caused the variance in performance across those 12,000 runs.
I would naturally assume that a highly complex, heavily mutated tumor would inherently cause algorithms to fail.
Most people would. But the actual biological features of the tumor, like how many mutations it had, only explained about 15% of the variance in performance.
Wait, really? Just 15%? That completely flips the script. I thought the tumor's physical complexity was the ultimate hurdle.
No, the choice of algorithm explained the variance considerably more. It accounted for between 19 and 35% of the variance.
So the algorithm you choose matters more than the actual biology of the tumor itself.
Yes. Meaning your clinical insight, the family tree you end up basing a critical treatment on, is dictated more by the software you happen to run than by the intrinsic biology of the patient's cancer.
That is frankly a little terrifying.
It is sobering. And across all these different algorithms, there was one critical technical metric that acted as the great filter.
Let's dig right into that, because if there is one core technical concept you take away from this deep dive, it is NRPCC, number of reads per chromosome copy.
Right. Usually in standard genome sequencing, we just talk about read depth. How many times the sequencer read a specific piece of DNA, like 30x depth, 60x depth, and so on.
But raw read depth is dangerously misleading in cancer genomics, isn't it?
Incredibly misleading. You have to adjust it for the tumor's purity because normal healthy cells dilute your cancer signal, and you have to adjust it for the tumor's ploidy.
Ploidy being how many copies of the chromosomes actually exist in the cancer cells? Okay, let me make sure I'm mapping this math correctly. If I have a tumor sample that is only 50% pure, meaning half the sample is just healthy tissue, my effective read depth for finding cancer mutations is instantly cut in half.
Right, you've lost half your signal right out of the gate.
And on top of that, if the cancer cells have mutated to have, say, 4 copies of a chromosome instead of the usual 2-like tetraploidy, A mutation that only exists on one of those 4 copies is diluted even further.
You've got it. NRPCC accounts for both of those dilutions. It is the ultimate measure of your statistical power to actually see what's happening in the subclonal architecture.
So it's a true resolution, and the researchers found a magic threshold for this, right?
They did. You need an NRPCC of at least 32 to reliably reconstruct a tumor's history. Below 32, the mathematical models just fall apart.
Why specifically does it fail, though? I keep thinking of it like taking a photo of a massive crowd at a stadium.
That's a good analogy.
If you're trying to categorize individual distinct faces way in the back, your NRPCC is essentially your camera's true resolution. Below 32 megapixels, the faces in the background just blur into static noise.
Exactly. And when an algorithm looks at that blurry data, it doesn't just throw an error, it confidently gives you the wrong answer.
Confidently wrong. The worst kind of wrong.
When the NRPCC is too low, the algorithms literally cannot mathematically distinguish real low-frequency subclonal mutations from random sequencing background noise.
So they just ignore them.
Yeah. Because they're designed to be conservative and avoid false positives, they tend to drop those low-frequency signals entirely. This causes a cascade of errors.
Right, because now the algorithm overestimates the tumor's purity, and it severely underestimates the total number of subclones.
It creates an illusion of simplicity. It looks at the blurry crowd and assumes it's just a few big, uniform groups of people.
Okay, let's recap this huge point. Algorithm choice matters more than tumor biology. NRPCC is the ultimate metric for sequencing power, and you need a score of at least 32 to see the true complexity of a tumor.
Spot on.
So listener, if you were designing a clinical trial today, think about this. Would you rather spend your budget sequencing fewer patients at a high 32+ NRPCC resolution or more patients at a lower, blurrier resolution?
It's the classic quality versus quantity debate in bioinformatics, but with actual human lives on the line.
Right. But what is truly fascinating is that this paper showed even if you have an infinite budget and sequence at a massive NRPCC of 100, the algorithms still get tricked.
They do. There are specific structural biological traps that break these models regardless of how good your resolution is.
And that takes us into the deeper mechanics. Let's look at trap 1, subclonal CNAs. How exactly do missing or extra chromosome chunks confuse the frequency math?
This is a massive blind spot for most models. Remember, algorithms use the variant allele frequency, the VAF, to decide how prevalent a subclone is.
Right, high frequency equals big old subclone.
Exactly. Let's walk through a timeline. Imagine an SNV mutation happens very early in a tumor's life. Because it's early, it gets passed down to almost all the cells, meaning it has a high cellular prevalence.
Makes sense. The algorithm should easily see a high VAF and say, an early dominant mutation, foundation of the tree.
But tumors are chaotic. What if, much later in the tumor's evolution, a new subclone emerges? And during a really messy cell division, that new subclone physically deletes the exact chunk of the chromosome where that original early mutation lived.
Oh, a subclonal loss. The mutation is just physically gone from that specific branch of the family tree.
Yes. Suddenly, when you grind up that bulk sample, the overall VAF, that early mutation, drops significantly because a huge chunk of the tumor cells no longer carry it.
And the algorithm doesn't know that.
Right. The mathematical model looks at that lower frequency, and because it relies on frequency to determine evolutionary timing, it wrongly assumes, oh, this low frequency means this mutation must belong to a smaller, much more recent subclone.
It severely underestimates the cellular prevalence because it has no way of knowing the mutation was historically present and then violently erased.
A brilliant trap.
It's like trying to count how many copies of a specific book were printed by looking for a unique typo on page 50. But halfway through the print run, the binding machine broke and just ripped page 50 out of half the books.
Yes.
You'd count the remaining typos, see a low number, and assume it was a rare recent misprint, completely missing the fact that the page itself was destroyed in half the population.
That is a perfect analogy. The algorithms assume frequency equals history, but subclonal losses break that rule entirely. And then trap 2 breaks the rules on a much larger scale, whole genome doubling, or WGD.
Right. We mentioned earlier how a tumor might double its genome. How does that practically scramble the algorithm's baseline math?
It comes down to overlapping distributions. In a normal diploid genome with 2 chromosome copies, a heterozygous mutation naturally sits around a 50% frequency.
Because it's on one of the 2 copies.
Right. And the algorithms use that 50% anchor to calibrate everything else. But if the tumor doubles its entire genome to 4 copies early on, mutations can now exist on 1, 2, 3, or all 4 copies.
Oh, wow. So you suddenly have multiple expected frequency peaks, like 25%, 50%, 75%.
Exactly. And when algorithms try to cluster the VAFs, these overlapping distributions create a massive statistical fog.
I can imagine.
The study found that in tumors with WGD, almost all algorithms fail to untangle this and frequently underestimated the true number of subclones. The complexity just overwhelms the models.
It's just too many overlapping frequencies to draw clean lines between the groups. But what about trap 3? This is a concept I found completely fascinating when I was prepping for this. Neutral tails.
Yes, neutral tails.
I imagine as a tumor grows, there's just a ton of background noise like passenger mutations that don't actually give the cancer a survival advantage. They just come along for the ride. Does high sequencing depth accidentally pick those up?
That is exactly what happens. As cancer cells rapidly divide, they naturally accumulate these harmless passenger point mutations. It's essentially the background ticking of the evolutionary clock.
And for a long time, standard sequencing depth was probably too low to even detect them, right?
Right, they were invisible. But if you push your sequencing depth incredibly high, say, to 128x coverage, suddenly this massive tail of extremely low-frequency mutations becomes visible in the data.
And I'm guessing the algorithms, which are mathematically designed to find patterns, look at this massive pile of new data and try to find a pattern that isn't actually there.
Precisely. They panic. The algorithms see thousands of these low-frequency mutations and assume they must represent new branches on the family tree.
So they force them into distinct clusters.
Yes, they predict spurious, fake subclones that don't biologically exist. They literally mistake the ambient noise of rapid cell division for genuine evolutionary branching.
But the paper actually tested a fix for this, right? You don't have to just accept the noise.
You don't. Through pre-filtering, they highlighted specialized tools, specifically one called Mobster.
Another great name.
Bioinformaticians love their acronyms. Mobster uses different statistical models, like Pareto distributions, to actually identify and model that neutral tail based on theoretical evolutionary dynamics.
So by isolating and filtering out those background passenger mutations before you hand the data over to the main reconstruction algorithm, you clear out the noise.
Exactly. At high sequencing depths, this pre-filtering step significantly improved the accuracy of the final family trees.
That makes perfect sense. Clear the static before you try to listen to the broadcast.
Right.
So looking at this whole landscape, we know certain algorithms are blind to subclonal deletions. We know others get overwhelmed by whole genome doubling, and some get completely distracted by neutral tails.
It's a minefield.
So if every algorithm has a specific mathematical blind spot, why not just combine them? Why not take all 31 algorithms, smash them together into a consensus model, and create a super algorithm?
It's the logical next step. And honestly, the researchers rigorously tested it. They built median and voting ensemble approaches, essentially taking the outputs of multiple algorithms and letting them vote on the final tumor architecture.
And let me guess. A majority rules vote doesn't work if the majority of the algorithms share the exact same blind spot.
You've hit the nail on the head.
Like, if 8 out of 10 algorithms can't handle a subclonal loss, the consensus is still going to be wrong.
Garbage in, majority rule garbage out. The ensembles didn't vastly outperform the single best state-of-the-art methods.
Really? No massive improvement.
Well, they did offer a modest improvement when estimating the total number of subclones, which was sub-challenge 1B, especially if you were flying blind and didn't know beforehand which single algorithm was optimized for your specific data set.
So it acts as a decent safety net.
Yeah, but running 10 different computationally heavy algorithms to generate one consensus tree costs a massive amount of cloud computing power, time, and money. It simply wasn't the magic bullet the field was hoping for.
Okay, time for our final quick recap. Subclonal DNA deletions trick algorithms. High sequencing depth reveals neutral tails that just add noise. And combining algorithms using ensembles is helpful, but definitely not a magic bullet.
That covers it.
So listener, knowing that subclonal losses literally hide mutations from our statistical models, how much of our current understanding of tumor evolution in the medical literature might actually be skewed by these invisible blind spots?
That is the exact kind of critical thinking this benchmark was designed to provoke in the scientific community. The models we've relied on for years have systemic biases.
It's wild to think about.
It is. But if we connect this to the bigger picture, the field isn't standing still.
Where is it going?
As long-read sequencing technologies, which read vastly longer continuous strands of DNA, become cheaper and more mature, we'll actually be able to phase mutations together physically.
So we won't have to rely purely on statistical frequencies.
Right, because we'll see exactly which mutations share the same physical strand of DNA. That physical linkage might solve these ambiguous phylogenies once and for all.
That is a huge leap forward.
But until long-read sequencing becomes the clinical standard, standard short-read sequencing remains our workhorse. And for that, rigorous benchmarking on realistic simulated datasets like the SNC Head Challenge is our only reliable compass to navigate these muddy waters.
So what does this all mean for you, whether you are analyzing this data, building these pipelines, or just trying to understand the bleeding edge of precision medicine?
It means proceed with caution.
Exactly. What this paper reveals is that subclonal reconstruction is not a single solved equation that you just plug data into. It is a highly sensitive, fragile pipeline.
The algorithm you choose dictates your results more than the biology itself.
And if you are doing this work, you must optimize your study design for that NRPCC metric accounting for purity and ploidy, not just raw read depth.
That's the biggest practical takeaway.
But I want to leave you with a final provocative thought to mull over. This massive 7-year simulated study showed us that our most sophisticated mathematical models are incredibly sensitive to their own internal design choices and statistical assumptions.
Extremely sensitive.
The way an algorithm handles a probability distribution fundamentally changes the shape of the biological family tree it outputs. It makes you wonder, across all areas of computational biology today, from predicting protein folding to mapping brain connectomes to designing personalized medicine, how many of our grand biological discoveries are actually just reflections of the algorithmic lenses we chose to look through?
Are we seeing the biology or are we just seeing the code?
Exactly. When we stare at the final data output, are we truly seeing the objective biology or are we just seeing the shape of our own code? Thank you so much for joining us on this deep dive. Keep questioning the data, and we will see you next time.